Hello friends, welcome to my channel Interesting World, and today we are going to see about an interesting topic of biocompatibility. Here you will see about in vitro test, animal test and usage test. First, biocompatibility test. There are three types of tests are used to analyze the biocompatibility test. They are in vitro, animal and usage test. First, in vitro. It is an extract of a material placed in direct contact with some biological term outside of an organism. Direct. It is the extract of a material directly to biological environment. Indirect. Contact involves a barrier such as agar, membrane filter or dentin. Conducted in cell culture, dishes, test tubes or other containers. Uses to measure cell growth, cell function, integrity of cells, genetic material. Disadvantage: limited resistance to clinical practice. An example: cytotoxicity using osteoblasts. Cell culture may be somewhat irrelevant if the material never contacts osteoblasts. This test can be used to check if a substance elected from the test material has general potential to be harmful to cells. Next we are going to see about animal test. It involves the placement of a material in an organism. Animals include for this test are rat, pigs, mice and monkeys. This differ from in vitro tests by animal is used rather than cells or tissues from an animal. Some animal tests, that is the material, are not applied as in patient. Example: when restorative material are implanted subcutaneously in rat, evaluated as toxic response. Other animal tests, they are materials are ground and feed to the animals, evaluated as acute or chronic toxicity. Therefore, these tests are becoming discontinued because of the evaluation of acute or chronic toxicity. Other animal tests involving guinea pigs, ability of a substance from a material to sensitive and elicit an allergic reaction is evaluated. Therefore, this test method provides reliable data that can be conducted to humans. Here, advantages are thereby interact with many complex biological systems to obtain a biological response. Disadvantages: high cost, control difficulty because of animals. Length of time may be required to obtain a measurable response. Tests are criticized by animal rights activists and other individuals because of concerned ethical treatment of test animals. Instead of these animal tests are essential for evaluating new materials prior to their clinical use. Next is usage test. Here, it is performed either in humans or animals. When a usage test is performed in humans, it is called as clinical trial. Choice of animals for a usage test is more limited than that of other animal tests because of size of anatomy of a given species. It used larger animals with anatomy that are similar to humans. Example: pulp. After placement of a restorative material is possible. Teeth are scheduled for extraction because of orthodontic reasons or normally obtained from young patients. These tests may have often open apexes which may react differently from old patients. Here disadvantages are design complexity, difficult to control experimentally, interpretation challenges, high cost, more duration, that is about 5 years. It may involve legal and confidential issues that do not occur in animal or in vitro tests. Thank you. We can meet in next interesting video. If you like this, please share and subscribe with your friends. Thank you.